Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Future Tech and Finding Genius podcast series. I have Lilach Sheiner. She's a senior lecturer on the Royal Society of Edinburgh, personal research fellow in parasitology. She's an expert in eukaryotic parasite cell biology, and we're going to talk about her research. So, Lilach, thanks for coming. How are you doing?
Yeah, very good. How are you?
Good. Yeah, I've started a series on parasitology. It's fascinating what parasites do. What about yourself? How long have you been working with parasites, and what fascinates you about them?
So I've been working parasites the vast majority of my career, all the way since my master's degree, which I probably don't care to say how long ago it was, but probably some good 13 years now. And what fascinates me is a couple of things. Firstly is the diversity of biology that parasites present compared to most of the biology we know. And you'll probably hear me mentioning that sort of concept again. And then the other thing, of course, many parasites cause diseases, and so understanding their biology could help prevent the disease by discovering new targets for intervention. So both of those things are huge motivations to study parasites.
So what do you mean when you say parasite biology is very different from, typical biology that we're familiar with?
So I think that's something that is a really it's a really important point that many people maybe fail to grasp or that many people are not aware of. And that is that, as mankind, we live in an environment that has huge diversity in it. And yet when we speak about what we know in terms of biology, most of what we know is about human and about animals, including, fish and worms and mice and other animals. But there is this huge sort of variety of life form that we know very, very little about, sort of how they operate, how they tick, how they do things differently to our cells, to our biology. And parasites are included in that sort of huge information gap.
So what are some specific examples of parasite biology that just really surprised you?
So let's look at the biology I'm focused on. So, human are composed of billions of cells. So the small operating unit in our body is a cell. And then parasites, the ones I'm studying, are also cells. And so within the cells, you have features that provide function, sort of like little compartments, like little rooms inside the cell. And one of those is known to many people as the powerhouse, the mitochondria, that makes energy for our cells in our body and also for the cells of the parasite. And so what was very surprising to myself and to the field as a whole is that parasite mitochondria operates very, very differently from human mitochondria. And that firstly teaches us about, again, sort of diversity in nature that we didn't really appreciate at the beginning. And again, brings up opportunities for drug discovery. So if you think about a parasite like malaria, for example, that many people know, and a drug like Malaron, that many people heard of, Malaron attacks the mitochondrion of the malaria parasite, but it doesn't attack the mitochondrion of the human. That's why malaron works. And this is because of these very sort of basic fundamental differences between how these two work.
So, all right. So what's an example? Tell me about, the fuel source that our human mitochondria use, and then contrast that with a parasite like malaria. What's so different about their mitochondria?
So what's different is it's more basic than the fuel source that they're using. The differences are in the active machineries. So, for example, mitochondria, both in human and in parasites, and in the malaria parasite, generate energy through this chain of complexes, so chain of little machineries that sort of pass molecules between one another, and in that way they generate this potential of energy. And the complexes, let's say the shape of the complexes in the malaria parasite is different to the shape of the complexes in human parasite. And that allows, for example, malaron to sort of fit nicely in the pockets of the complexes or the machineries in malaria and not fit in the complexes of the human mitochondria.
Okay. So different shapes of receptors, let's say on the surface of their mitochondria, possibly different mechanism, but different enough that you can have drugs like malaron that again would destroy their mitochondria, malaria, but not ours. That's good.
Yeah, exactly. So different active machineries. So they're not really receptors, but I don't know if that matters very much. They're they're active complexes or enzymes, you can say. They're sort of these big enzymes. Okay,
so what parasites are you studying now specifically? Are you studying malaria or Toxoplasmosis?
Yeah, so I do. I study two types of parasites. One is the parasite that causes Toxoplasmosis, and the other is the parasite that causes malaria. And the thing that maybe not many people know is that those parasites are biologically very similar to one another. And Toxoplasma allows us to do certain types of experiment that maybe the malaria parasite is a little harder to do with. And so that's why we use Toxoplasma as a model in many cases to study to ask the research questions we're asking about the parasites.
Well, again, what are you trying to figure out about Toxoplasmodium? Well, first of all, let's back up. Where does it come from? What kind of host does it go into, and what does it do once it's in there?
So Toxoplasma infects a whole range of animals, and that includes human. And the disease that it causes is called Toxoplasmosis. And many people who've heard of Toxoplasmosis or Toxoplasma would associate it to this parasite that you might get if you clean your cat litter and don't wash your hands, and that can make your behavior a bit funny. And so that's what Toxoplasma is most famous for. In fact, the most common way to get Toxoplasmosis is from undercooked meat and from unwashed vegetables. And the sort of health risks that are the best studied one, rather than the behavioral one, are the ones that Toxoplasma could cause severe brain damage. And that typically only happens in people whose immune system doesn't work. For example, AIDS patients or people who are about to have a transplantation and their immune system is suppressed so that they accept the new organ. And that's also why Toxoplasma and Toxoplasmosis are involved in pregnancy, because babies don't have their immune system before they're born. And so if mom gets Toxoplasma for the first time during pregnancy, then the baby is at risk for brain damage. But mostly healthy people could get infected and then just have a little bit of flu-like symptoms and then ride off the disease.
Once someone has the Toxoplasmodium, does it stay forever, or what can they do?
So Toxoplasma is is thought to persist in your body for the rest of your life. And when the immune system attacks the parasite, it goes into this dormant form that stays forever. And one of the biggest issues with treatment for Toxoplasma and Toxoplasmosis is that we don't have a cure that could clear the chronic infection. And so there isn't much you could do. What doctors tend to do if somebody is about to become immunocompromised because they have a disease that is known to interfere with the immune system, then doctors would test for positivity or negativity with Toxoplasmosis, and then they know to expect a potential outbreak when the patient becomes immunocompromised. And then for the form of the disease that causes the acute infection, so when the dormant parasite outbreaks again, for that we have drugs like pyrimethamine, for example.
Well, how long if Toxoplasma can stay dormant in someone forever, what would activate that parasite later on that would either harm them or cause them to spread it to someone else?
So human to human, there is no spread of Toxoplasma. So the way to get Toxoplasma infection is by eating undercooked meat or unwashed vegetables. And so there is no risk for human to human infection. And what typically triggers the disease is if you go immunocompromised. So once your immune system is off guard, that's when the parasite can sort of wake up again and cause symptoms.
Huh. So, God forbid someone needs an organ transplant when they're older. They've had this, or they have cancer and they get chemo. Let's say they need to be immunosuppressed. They might not be able to be, right?
So they would be able to. What the doctors would typically do is test if they are seropositive for Toxoplasma, and if they are, then they keep testing whether the parasite became undormant or whether the parasite woke up. And then there are treatments that you can give when the parasite is awake again. And things like pyrimethamine, for example.
So what specifically are you trying to figure out in regards to Toxoplasma?
And so, like I said, my sort of main motivation to study Toxoplasma and other parasites is not so much to understand something about the disease or about the parasite, but more to enrich our understanding of different life forms. So I don't know if that's a good enough.
No, that's fine. With parasites, what's interesting to me is that they're able to inhabit sometimes multiple hosts, an intermediate host and then like a definitive host. And they can be completely different creatures. And then they can go into dormant states and then come out of them a long time later. So they seem to have like very malleable, sophisticated biology. So in those instances, are there specific things about, parasite biology that you want to study to see how other creatures might be able to do that or how we can learn from them? Like, again, their ability to be in different biological hosts and are completely different biologically. What is it about them that you want to figure out?
So I think what's interesting about understanding how parasites work and how a parasite cell work in a different way to a human cell is that it gives us tools to later on make sort of biotechnological progress. Because parasites have such a unique and complex biology, there is a lot we can learn from them and sort of take advantage of that. And so one example I could give you is work that we've done recently. So Toxoplasma has the ability to cross the blood-brain barrier. And typically things don't get to the brain. The blood-brain barrier is very selective to try to protect the brain from anything getting into it. And this is a big issue in brain therapy. And so what we've done recently is we've generated Toxoplasma parasites that express human proteins that could then go into the brain the way they do when they during infection, and then they can deposit the human protein back in the brain. And so basically we're trying to use Toxoplasma to turn it into a therapy delivery. And of course, there is a lot of work we need to do to reduce the infectivity part or the disease-causing part of the parasite. But by understanding how these parasites live, we got to even think of this idea of using them as couriers. So I think that's a really good example of the sort of things we can learn when we study life forms that are very different to the one we already know.
Okay. In terms of crossing the blood-brain barrier, I don't know, has it been literally observed? I mean, I guess you'd find parasites, across the blood-brain barrier. Is it possible in a lab to literally set up a situation where you'd culture cells and watch a parasite do such a thing?
Yeah, so that's
be observed.
Yeah, so that's been done. Observing parasites crossing the blood-brain barrier has been done by some researchers in mice models, and you can really see that sort of in an experimental setting.
Okay, so with that observation, what's been observed? I mean, are they burrowing through the barrier? Are they releasing a chemical that loosens the bonds of the barrier that they fit through? Are they sending through maybe an emissary or a part of themselves that grows another parasite? Like, what's the mechanism?
I see. So, so how the parasite crosses the blood-brain barrier has been studied in different types of model system, and in some cases really in animal models, and have been really observed in live microscopy. And there are sort of three different ways that are accepted at the moment that the parasite crosses. And so the first one is that the parasite could sort of piggyback on cells of the immune system that do get access to the brain. So it goes into those cells of the immune system and then just enter like a Trojan horse. And then the two other ways are both dependent on the parasite motility. So these parasites are very motile. They have a little sort of walking machinery almost, or gliding machinery it's called. And then either they penetrate the cell and come out on the other side, or they really invade the cell and then multiply and burst out of the other side.
Yeah. So they'll invade cells in our body that can go through the blood-brain barrier, put material in them, the cell migrates through, and then toxoplasma are formed inside the cell using the cell machinery and then explode out. Is that the mechanism?
So that's one of the ways that Toxoplasma is thought to move through the blood-brain barrier is by going into an immune cell that can cross the blood-brain barrier, and then after the barrier, the parasite comes out of the immune cell. So that's one of the ways that it could go through.
So in one way, they act kind of like viruses.
I guess there are some similarities between Toxoplasma and Zika virus. That's in that sense, yeah. So
what are you hoping to figure out that you think will be very useful in defending against Toxoplasma or other parasites? Like, are there any hypotheses you have that you're working on experimentally?
Yes, so both Toxoplasma and Plasmodium, which causes malaria, have in their mitochondria a little machine that makes all the proteins, all the building blocks that are necessary for the mitochondria and therefore for the parasites to survive. And this machine, it's called the ribosome, is also present in human, but the parasite one looks and acts very, very differently to the human one. So if you saw a picture of the mitochondrial ribosome in parasites and the mitochondrial ribosome in human, you wouldn't think they are the same. They're doing the same thing. And so this presents opportunities for drugs to interfere only with the parasite machinery and not with the human one. And so what we're hoping is to understand the structure and the function of this machine to enough details so that we could then design drugs stop the parasite machine and not the human one.
Okay. Any other parasites that you observe that have even more abilities than the ones we talked about? Are there any super parasites out there that just are unbelievable in what they can do?
Yes, I think. What's a good example for that? Yeah, I'm not sure I have a great example for that right now.
Okay. Are there any? Are there other parasites that you've studied that have just very different biology to the ones that you're even currently studying? Like, how diverse are the forms and abilities of parasites in general? Are they very diverse, just like animals are or other creatures are, or are they pretty similar?
So different parasites are extremely different from one another and from the cells of animals and human. Some example, actually, from the parasite I am working on is that they have a plastid. So plants, for example, have a plastid. They do photosynthesis. And parasites, some parasites also have a plastid. Obviously, they don't do photosynthesis because they're inside another animal. So they don't have access to a light, to the sunlight. But then there are other sort of metabolic pathways that are hosted in those plastids, and so in a way you could consider that being a super cell because they can make themselves their own food in various different ways. I don't know if that's
really so they can make their own food. Like animal cells would, and they can also do it how plant cells would. You mean?
In a way, yeah, I think that's a way to summarize it. They can make themselves at least some of the things they need to survive, in two different ways, as well as they can scavenge some food from the host where they reside. So they cover their chances to survive on various different ways, unlike human cells.
Wow. What do you think drives this hyperability of parasites versus other creatures? I mean, do they seem to adapt faster than other creatures, or?
I think the issue is niches, and so where do different organisms live? And these parasites have to survive immune systems of their host. They have to survive. They have to find a way to move between one host and another, so to transmit. Sometimes they have to survive in the environment until they get picked up by the next host. And I think those needs push them to have all these sort of new and exciting biological solutions that cells of multicellular organisms maybe don't need to deal with.
Okay. So any breakthroughs that you think you're close to, in the next year or so?
So we have stumbled across some compounds that are much more potent to the parasites than to human cells, and so we are following up on those as potential leads for drugs for Toxoplasmosis and for malaria, and probably considering the drug discovery process, that wouldn't happen next year, but it's certainly looking very exciting at the moment.
Well, very good. What's the best way for people to learn more about your research and to get in touch and about parasites in general?
The best way would be to go on my website, which is lilachsheiner.wixsite.com, or just to search Sheiner Lab on Google. And I'm also on Twitter at Sheiner Lab.
Okay, well, very good. Well, Lilach, it was great to speak to you, and I appreciate you coming on.
You too, and thank you for taking the time to ask me some cool questions.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And be smarter than everybody else? Become a premium member at findinggeniuspodcast.com.